This week we're going to be taking a look at chapter 4 in the textbook, which is specifically on bacteria. But I actually want to start out with an introduction to cells, and I'll talk about why that is here in just a second. So we'll start with cells, and then in this first video for this week, and then in the second video we'll focus more specifically on the bacteria. So why I'm focusing on cells is that cells are often defined as the fundamental units of life. Fundamental meaning if you try to break something down below the level of a cell, take a cell apart, you'll no longer have the minimum requirements of life. And we'll get to what we mean by life here in a second. Turns out it's a little more difficult to define than you might think. But all living things are made up of at least one cell, and one is the minimum. However, complex life forms like you and I will have literally tens of trillions, with a T, trillions of cells. So the spectrum of life would be at least one cell up into the trillions of cells for a single species. So part of biology is really focuses on the cell, given that it's this fundamental unit of life. Now, cells have so much diversity that we won't be able to cover that even given the entire 16 weeks of the class, and certainly not in this one video. So I'll try to keep it as broad as possible, and then we'll focus in a little bit more on some of those specific types in the next couple of chapters. But when we look at cells, their structure and function can be kind of categorized along with complex organisms in relation to the terms tissues and organs. Okay, so and if you've had anatomy and physiology, or maybe you're taking that right now, these terms come up more often. So multicellular organisms like you and I don't consist necessarily of just cells. Those are the fundamental units, but cells group together to form tissues, which are collections of all the same kind of cell, and then multiple tissues form to create organs, defined here as groups of tissues that form a specific function. So for example, you will have cells in the lungs that make up specific types of lung tissue, and then various forms of lung tissue collectively make up the lungs, which are part of the respiratory system. So very often complex animals consist not only of just cells, but complex networks of cells. Now, as we go forward in the next couple chapters, we'll focus mostly on microorganisms. So I'm putting those terms a little bit out of context just to kind of give you an idea of how those tend to apply to animal life, humans like you and I. Now, the next part here is going to be a kind of a list of the characteristics of living organisms. So, as I said before, it's actually kind of difficult to say what life is. This is actually something that's debated, so keep that in mind with this list. This isn't necessarily a universal list that everyone agrees upon. But when we try to look at what life is, in no particular order, these are not ranked necessarily by their highest priority. Life at the cell level, or a bit above the cell level, such as the tissue or the organs, for example, always has the ability to respond to the environment. Unlike a rock or inert soil, it can pick up on feedback and even send signals in response to that. One of the most miraculous, underappreciated parts of life is that it has the ability to reproduce. Single-celled organisms can simply make copies of themselves and duplicate rather quickly in many cases. More complex organisms reproduce through sexual reproduction. But that ability to copy and propagate life is probably one of the most miraculous parts, very often unappreciated. Adaptation, part of the evolutionary process, is the way that life responds to the environment to become more fit to that environment. So life tends not to stay the same. It tends to change in response to the environment, and that's what we call adaptation. Another key part is energy use. So energy is crucial at every aspect of life in order to maintain life and to help it respond, to help it reproduce, to help it adapt. So energy is just part of the list here. I won't go into the details of energy, but that is a key part of it as well. And we will have a little bit more about that in Unit 2. excuse me there. Another part is that they exhibit homeostasis. Now, homeostasis is sometimes, if you're not familiar with that word, kind of a mouthful there. But homo means to stay the same, and stasis is kind of a balancing act. So you're kind of staying the same through a balancing. That's not the definition, but that's sort of a way to think about it. So homeostasis means that you're going to maintain an equilibrium, typically in reference to the cellular environment. That could certainly also apply to the tissue or the organ level as well, but you're trying to maintain a balance. The best way I think about it, to simplify it, is it's kind of like bringing in groceries to your house, which is part of the energy resource acquisition, but then also taking out the trash. If you didn't do both, you would not be able to maintain a balance in your home. You need energy and food and resources, but those energy, food, and resources also accumulate waste, and so you have to take that waste out as well. Cells do the same thing at the microscopic environment, obviously in a very different way, but that's probably the easiest way to think about homeostasis, bringing things in, taking things out in order to maintain a balance. Hereditary information is always found in life. Another very interesting part of life is that it contains information. And that information is something that we'll talk about when we get to genetics. Genetics, which is chapter 6, it'll be out of sequence with the text, with the way I cover it, but genetics and hereditary information go hand in hand. So this is essentially DNA and also RNA. So all life has it, and there's actually no exceptions to that. And what's fascinating about it, as we'll talk about, is that it's very, very consistent across life. Growth and development. This is actually different than reproduction, although it would be understandable to get those two mixed up. Reproduction is the act of sort of propagating new life from existing life, whereas growth and development has more to do with a single organism changing as it ages, also different from adaptation. So you see this very clearly in humans. You start off as an embryo that then develops into a fetus, and then an infant, and then a toddler, etc. And so that's kind of an example of growth and development. Not every organism obviously does that in the same way, but there is always a process of growth and development in some capacity. And the last thing, which is kind of a segue to the next part, is that they always consist of one or more cells, as I've alluded to. So life has to have at least one cell, and many life forms have significantly more than one cell, but that is always the minimum. So that's kind of the laundry list there of your characteristics. And as I mentioned, those are not universally consistent. So those are kind of that's the list that I've put forward, and you will see some variation depending on your source. Now, when we look at the cells, we want to start with the cell types, and these are very, very broad. So interestingly, you can actually take every cell on Earth, no matter what kind, and loosely lump it into one of two broad categories. And so these are prokaryotic and eukaryotic. You may be familiar with these terms. They come up quite a bit, at least in biology. And so prokaryotic kind of means the first cell, and eukaryotic roughly translates to the quote-unquote true cell. And those are just sort of the throwback terms that they coined early on in the study of life. Prokaryotic organisms in this class will largely emphasize our bacteria. So bacteria are considered a prokaryotic life form. These tend to be quote-unquote simple. I say quote-unquote because simple is a relative term. But bacteria don't have the complexity as, say, an animal cell, or even a plant cell for that matter. So that's kind of the idea of prokaryotic, sort of simpler, the earlier types of cells. Bacteria being the most significant, certainly to this class and the emphasis in this class. Another one that you want to at least be familiar with is what's called the archaea, archaea as it's sometimes said. These are interesting, less relevant to our everyday life, but another example of a prokaryotic, sort of simpler type of cell. So at the very least for this class, you want to be familiar with this list of what are the two types of prokaryotes. Eukaryotic organisms are, in essence, anything that's not a bacteria or archaea would be a eukaryote. And so that would include all the plants, all the animals, fungi, and then protista or protists. Most people are going to be familiar with the terms plants, animals, and fungi. Protista, probably not as much, but protista are just sort of small, very often single-celled eukaryotic organisms. and we will touch on those, by the way, in chapter 5. Now, a little bit confusingly, these two broad groups of cells are actually divided into, divvied up into what are called the three domains of life. And as a real quick backstory, there actually only used to be two domains of life, which were the prokarya and the eukarya. Today, what they've done is they've divided the bacteria and the archaea into their own domains, along with the eukarya domain. So your three domains are the bacteria domain, the archaea domain, and the eukarya domain. Okay, and there's a reason why that is. I don't want to spend a lot of time going into that, but those are the three domains, and ultimately, maybe a little more specifically, all life can be categorized in those three domains, just depending on the context. So, life consists of cells. Those are the broadest ways that we can categorize cells. So now what we're going to do is look at the minimum features of life, because if we tried to look at all the features of life, it wouldn't be able to do that. You'd spend your whole life looking at that. So these are the minimal features, meaning like you can't take any one of these away and still have a cell. So I mentioned hereditary information as being one of the characteristics of life. So DNA and RNA both constitute this hereditary information. We often think more about DNA, and that's actually the one that's listed here in this figure, but technically that includes both. The cell membrane, more technically referred to as the plasma membrane, depending on who you're talking to. This is the distinct boundary of a cell. And a cell is essentially a unit that is segregated from the rest of its environment by this membrane. So it creates a distinct boundary. And we will have a separate lecture on that in the next chapter. That'll be part 1 of chapter 5, where we'll go more into the characteristics of just the cell membrane. Ribosomes are another interesting part. Don't spend as much time on those. They will come up later in the semester because they interface with our DNA and RNA. So they come up when we talk about genetics. But ribosomes, for now, just need to be recognized as the protein synthesizing units of the cell. So as we'll learn later, all life requires protein. And in order to get protein, you need ribosomes. Without them, you cannot build protein, and therefore life is not possible without them. And the last one is sort of just an attribute to the fact that life requires water and other dissolved elements within the water. And that water and those dissolved elements are found at a minimum inside of the cell, within the cell membrane. So once you kind of take the water and all of the things that are dissolved within it, that collection becomes what's referred to as the cytoplasm or the cytosol. Once again, context is kind of depending on who you're asking or who's talking about it. Both terms more or less mean the same thing. So that's sort of the gelatinous, liquidy part that sort of makes up the internal aspects of the cell. So 4 minimum features, all life has them. There are variations to them, but you cannot have life without having at least those 4 things. Now, when we look at DNA, I'll give you a quick rundown of DNA and then later a chapter that comes along with that. I think DNA is important to understand, so I'll give you a quick rundown of it. DNA stands for deoxyribose nucleic acid. You'll want to know what that term means, but it's not critical that you memorize that for the rest of your life. But that's what that actually stands for, deoxyribose nucleic acid. When I said earlier, DNA and really hereditary information is information, it is essentially an alphabet made up of 4 letters. Now, we use our own letters, A, T, C, and G, but what these actually are chemicals. Adenine, thymine, cytosine, and guanine, as we'll learn later. So it's a chemical alphabet that is used as a way to store and transmit information in the cell. So that's essentially the nature of DNA. When we start putting the letters together into combinations, like you would put letters together to form words and sentences and paragraphs, those minimal sort of combinations are what we refer to as genes or gene segments. And you've probably heard that term before, that you hear it somewhat commonly in just everyday conversation, but genes are essentially the units of information that kind of spell out the characteristics of life. Kind of like the way letters make up words and sentences. So one letter by itself rarely means anything, but combinations can spell out valuable information. That's kind of like what a gene is a combination of letters that ultimately give the cell information, and very often that information relates to the characteristics. So when we go back to proteins, I said ribosomes build proteins. Proteins are essential for life. Proteins are actually the product of the gene information. So in the cell, the gene is actually more like a recipe that tells your body how to build specific proteins. And those proteins then ultimately conduct the functioning at kind of the minimal level of the cell. That more comes up specifically in Unit 2. We'll talk in a decent amount of detail about how that works, but that's kind of the quick rundown here. So you might also think about DNA like a blueprint. There are different ways of analogizing it. A blueprint is a pretty good way to think about it. So it kind of gives your body the information for design and functionality. The proteins are kind of the activated downstream product of that information. So if you just have, let's say, a blueprint stashed away in a book on your bookshelf, it's not doing anything. But once you pull that book and the blueprint off the shelf and then you read it and start building, that's kind of like what a protein is. It's the product of that information in action. So that's kind of how genes and hereditary information ultimately become sort of the cellular process. The mechanism is often referred to as gene expression, kind of like you have an idea in your head, information, and then you say it out loud. You're expressing your idea. That's what we often refer to as gene expression. That's the process by which the information is converted into a product, which is very often, not always, a protein, which then takes an effect. Epigenetics will come up in the genetic section. Epigenetics is a newer area of genetic research that has to do with the kind of control of gene information, almost like an on-off switch that tells the cell when to activate a gene or when not to activate a gene. So gene expression, in just a little bit more detail here, begins with DNA. DNA then is transcribed, converted into RNA, more technically what's called mRNA, which stands for messenger RNA. And then it's the messenger RNA through the ribosome, although it's not labeled here, this is representing a ribosome, that takes the mRNA and turns it into a protein, which is essentially a product of material synthesis based upon the information that ultimately begins with the DNA. Much like a blueprint becomes a physical product, gene expression begins with DNA and ends in protein. That's where ribosomes come in. They are the key part of that building of the protein towards the end of gene expression. So the blueprint is the kind of broad analogy for that process. Now, a term to summarize that process is referred to as the central dogma of biology. Central means that this process occurs in all of life. Dogma is kind of an interesting way to describe it. Dogma is sort of like a belief that you don't question or you don't have any reason to question, sort of like dogmatic thinking. So the idea is that this has been studied long enough now. It's been investigated to the point that the dogmatic assumption is that all of life operates this way, because so far there has yet to be a single example that has ever been found where a life form operates outside of this process. So that's kind of what that term implies. It means that every life form on Earth, with not one exception, has DNA as the foundation of its hereditary information, converts that DNA into RNA, and then ultimately into protein, where the functionality of the cell kind of begins. So that's kind of the idea of what that term means. It is key to all of life and yet to have a single exception. So this little diagram down here will show up on quizzes and tests, something like which of the following best describe the central dogma of biology, and you'll just want to recognize that flow. Starts with DNA, DNA becomes RNA, RNA becomes protein. That's a simplified way of putting it, but that's the easiest way to think about it. So that's just kind of a quick rundown more on the hereditary side of things. Now when we get into protein functionality, we're looking at really the building blocks of protein first, and then the complex structure that arise from the building blocks being put together in a highly specific way. So proteins are ultimately chains of amino acids. You can see here in this figure, each one of these little circles represents an individual amino acid. The colors indicate that there are different kinds. They're linked together into a long chain, and then that chain will start to fold into some kind of, very often, three-dimensional pattern. You don't really see that with these figures here, but it ultimately will become a three-dimensional figure. So the folding is a result of interactions between the amino acid chain, and ultimately the folding is a product of the genetic information. Going back to what I just said about the central dogma, the way that the protein comes together is directly a product of the information in the DNA to begin with. The DNA literally spells out exactly how to build the protein, one amino acid at a time, and depending on what sequence of amino acids you put together and how long the chain is, you'll ultimately get different folded products, different proteins that have different functions depending on the exact folding process. So you will end up at the end of a protein synthesizing event with a three-dimensional structure. And the three-dimensional structure will ultimately determine the function of that protein. So genetic information spells out amino acid sequence. Amino acid sequence determines protein folding. Protein folding determines protein function, and thus the viability and characteristics of all living organisms. The only thing that's different from one form of life to another is just the types of proteins that they make. Not whether they make proteins or not, or whether they use DNA to do it, but just what does the DNA information say, and how do the proteins fold differently. That's pretty much the essence of how life differs. You can see a little bit better here how the process of gene expression unfolds, and then you'll end up with a sequence of amino acids. Those start to fold into various shapes, and then you'll ultimately get kind of a three-dimensional structure here that allows that protein to interface or interact with other molecules, and very often in what's referred to as lock and key interaction. Lock and key interaction is a way of kind of describing how the shape of a protein is intended to form its function. So let me give you some examples. One of the most important, arguably the most important type of protein are called enzymes. You probably heard that term. Enzymes are just a specific category of protein. And so ultimately, they're the kind of the kind of protein that drives our metabolism. So everything from building muscle to breaking down your food to create energy is all dictated by enzymes, many of them. So you may be familiar with this one. Lactose is your milk sugar, and lactase is the specific enzyme that breaks down lactose. And usually, but not always, they're named in some way based upon what they interact with. So lactase, the enzyme, lactose is the substrate that the lactase enzyme acts upon. So if you're lactose intolerant, you have some issue producing lactase, and as a result, you get kind of a buildup of lactose byproduct, and you get usually some kind of indigestion. Another similar example is sucrose and sucrase. Sucrose is your sugar, typical table sugar, and sucrase is an enzyme that breaks that down. Other examples of where proteins are very important is with immunity and infection, or in other words, immunology. You've probably heard this term as well, antibodies. These are a specific type of protein that relates to immune function. Much like the enzyme, antibodies all have a very specific shape. And the shape in this context determines what kind of pathogen, or even more specifically parts of a pathogen, the antibody will interact with. And that will come up towards the end of the semester in the last unit. Every kind of pathogen, like a virus, has its own shape. And ultimately, different antibodies are required to interact with different types of viruses, different types of bacteria, any kind of pathogen in general, and very often down to the specific part of the pathogen. Each antibody can have a different interaction because of its shape. So ultimately, what our body does is it creates a whole variety of antibodies with many different shapes so that we can interact with and neutralize many different types of harmful species as I mentioned in the first video, viruses tend to be the most common pathogen that we interact with. Another example here is the way our cells communicate. So cell-to-cell communication is another aspect of protein function. All cells have what are referred to as protein receptors. This is usually part of the cell membrane. They're little, almost like studded structures that stick out from the cell with their own shape that very often act as like a signal receptor. So the shape allows it to grab a molecule that is acting as a feedback signal that, when binding, gives an internal message or sense of feedback to the cell, causing it to respond in some way. So ultimately, all cells communicate through some form of protein signaling process. The exact nature of it will vary, but ultimately the protein acts as a feedback mechanism that picks up signals to give the cell information about its environment. So those are some examples of how proteins work and some of the key aspects of why they're important. A couple more things on cells and we'll wrap this up here. Cells, as you already kind of know, are very small. You don't ever really see a cell with your naked eye. And we'll get to why that is here in a second, but just to put some numbers to it, an inch is 2.5 centimeters, 1 centimeter, 10 millimeters, 1 millimeter is 1,000 micrometers, okay? So 1 millimeter, if you were to pull up a metric ruler, is the smallest notch on a metric ruler. 1,000 micrometers or 1,000 microns can fit across that tiniest little space on a metric ruler. That's the kind of units of measure that we typically use to measure cells in a general sense. You can then go even smaller if you're going to measure like the size of a protein or a specific subcompartment of a cell. And then you can take that one little micrometer, which are 1,000 of those fit into a millimeter, and for every 1 micrometer, you have 1,000 nanometers. So a nanometer is the smallest unit of measure that we typically use, and that's when you start using that measure to measure some specific internal part of a cell, like the size of a protein, for example. So 1 inch is approximately 25,000 microns, or 25 million nanometers. So when we start looking at some of the dimensions here, you'll see that those types of numbers are necessary to kind of understand what we're measuring. So as I kind of mentioned, most cells are undetectable. You could probably imply this, but to state it specifically, they require a microscope to see them. And even then, it's not always that you can see everything with a microscope. There's lots of different types of microscopes, but microscopes in some capacity are almost always needed to see a cell. So when we start looking at some of these numbers here, prokaryotes are usually a fraction of a micrometer, anywhere from 0.2 to 2 micrometers. So if we go back to this previous slide here, we can see that 1 centimeter has roughly 10 millimeters and 1,000 micrometers, so roughly 2,000 micrometers. And so if a prokaryote, even a big one, is 2 micrometers, we can fit 1,000 or so bacterial cells across that 1 millimeter range. So just in a tiny little space on a ruler, you can fit potentially thousands, if not more. That's on the big side. So you can fit potentially thousands and thousands of prokaryotes just in that tiny little space. And that's why we can't see them. Our eyes just don't have that kind of resolution. Now, eukaryotes are on average bigger, 10 to 100 micrometers, but still typically much too small to see with the naked eye. So by comparison, they're bigger, but still relatively small overall. And remember that eukaryotes are the more complex types of cells. When you look at something like a virus, Viruses are typically measured in these nanometers, and so viruses are therefore usually much smaller than a typical cell. I will say that viruses range in size. They're all over the place in terms of their size. There are very large viruses that have been discovered, but your typical virus is in that sub micrometer category, or in other words, nanometers. So you're typically measuring them in the smallest units possible. And then proteins, as we talked about earlier, are usually even smaller than that, 3 to 10 nanometers. So even with a microscope, especially like the type that we use here at the campus, you cannot see a virus with that type of microscope. There are some that allow you to see them, but most microscopes you can't see a virus. And as far as I'm aware, there's no microscopes that allow you to see proteins. And the only reason I say that is because technology changes, but as far as I'm aware, there's no microscope that will allow you to see a protein. Proteins have to be studied in ways that kind of go beyond microscopy. So that's kind of an idea of the size of which, when we talk about micro, small, that kind of puts some more logistical parameters to what that means. Last thing here, and we'll wrap this one up, why are cells so small? Because you might think that given enough time, cells could adapt and change and grow bigger, and maybe there would be some survival advantages to that. But the reason why cells remain small has to do with this specific logistical aspect, which is to maintain proper surface to volume ratio. So if you only remember one thing from this slide, remember that first line. Why are cells small? To maintain proper to surface volume ratio. So if you go back to what I said earlier about homeostasis, I mentioned bringing in the groceries, taking out the trash. The bigger your house is, as an analogy, the more space you have to travel. If your house is huge, you have to walk further to your refrigerator and walk further to your trash can. Now imagine that every day that you do that, you're on a timer. You have to get the groceries in less than 10 seconds, and you have to get the waste out in less than 10 seconds, just as an analogy. And if you don't do that, you die. That's kind of the nature of a cell. It's not a sort of arbitrary time frame. Cells have to get nutrients in immediately, and they have to get waste out immediately. There's not much of a time frame that they can delay that on. So what we find is that if you try to get a cell too big, the ability to absorb nutrients and release waste becomes so inefficient that the cell actually dies. And it may survive for a period of time, but as soon as something becomes critical, like stress or an environmental stress, then they can no longer survive. So that's a more specific reason why. You have to maintain homeostasis, and the bigger you are, the more difficult that becomes, and at some point it becomes impossible for survival. So ultimately, what life does when we look at multicellular organisms and large life forms like you and I, or an elephant or a blue whale, to take an extreme, what's happening in that situation is not that the cells themselves are getting bigger. The cells will always max out at a certain size, and there's a lot of nuance to that, to be honest, but there is a certain maximum capacity for size. So as an organism gets bigger, it simply adds more cells to the collection. Each cell still functions as an independent unit, more or less, but the unit or the collection rather, grows in multitude, not individual size. So that's ultimately how life becomes bigger and bigger and bigger, is by adding to the collection, but not by changing the individual unit of the cell itself. All right, as always, email me if you have any questions, and the next video this week will focus more specifically on the bacteria in reference to the chapter this week, which is chapter 4.